The adult mammalian brain physically reorganizes its cellular architecture to prepare for the severe physiological demands of motherhood. At the center of this structural transformation is adult hippocampal neurogenesis, or AHN. AHN is the continuous biological mechanism where neural stem cells actively proliferate and differentiate into functional, mature neurons long after early brain development has concluded. This generation of new cells is heavily localized to a precise anatomical region, the subgranular zone, or SGZ, located within the dentate gyrus of the hippocampus. The SGZ is a narrow layer of cells sitting between the granule cell layer and the hilus, serving as a primary hub for learning and memory circuits. Within the SGZ, neural stem cells continuously multiply, providing a persistent source of new cellular material even in a fully mature adult brain. Rather than operating as a steady, static background process, this neurogenic niche is highly reactive. It waits for specific chemical signals to either accelerate or halt cellular production. The neurons produced in the dentate gyrus directly integrate into networks that govern spatial navigation, new memory formation, and emotional regulation for maternal care. To prove that these maternal behaviors rely strictly on neurogenesis, researchers face a difficult methodological problem. How do you isolate and measure the behavioral impact of newly born cells without damaging the rest of the existing brain? Researchers track how a surviving cell actually matures and wires itself into the active hippocampal circuit. The life cycle begins at the neural precursor stage. Researchers track these early cells using BRDU and Ki67. BRDU and Ki67 are specialized biochemical markers that integrate into the DNA of actively dividing cells, making them visible under microscopic analysis. If the cell survives this initial phase, it enters migration and differentiation. It becomes an immature neuron, identifiable by the presence of D6. D6, or double cortin, is a microtubule-associated protein exclusively expressed by developing neurons as they extend their early dendrites. In the final stage, the cell fully integrates into the local circuit as a mature, functioning neuron. At this point, it expresses nu-N. Nu-N, or neuronal nuclei, is a standard nuclear protein biomarker indicating that a neuron has permanently exited the cell cycle and reached full maturity. A newborn cell's ability to successfully navigate this BRDU to nu-N maturation pathway dictates the extent of the brain's plasticity. During pregnancy, this entire cellular journey is strictly governed by dramatic shifts in gestational hormones. At the onset of mammalian pregnancy, the female physiological system experiences a massive sudden influx of reproductive hormones. This chart shows the parallel rise of specific endocrine levels throughout a standard 40-week gestation period. The lines representing estrogens and progesterone climb steadily in tandem as the pregnancy progresses. This steady accumulation abruptly reverses at parturition, or childbirth. Both estrogen and progesterone concentrations crash within hours of delivery. Simultaneously, the system introduces a sharp counter-movement. Prolactin, a peptide hormone primarily responsible for stimulating milk production, spikes exactly as the other steroids plummet, remaining elevated throughout lactation. This precise neuroendocrine intersection, where estrogens drop and prolactin surges, creates a highly volatile chemical environment. This specific hormonal volatility ultimately dictates the survival rate of the newly generated neurons. This table displays the effects of reintroducing estrogen to these subjects over varying timeframes. During acute exposure, lasting just minutes to a few hours, the data shows a sharp, immediate upregulation in cellular proliferation within the hippocampus. A standard clinical estuntion follows this data. If a brief pulse of estrogen creates a burst of new neurons, then maintaining high estrogen levels throughout a long pregnancy should produce massive neural growth. The data contradicts this. Under chronic exposure lasting several weeks, cellular proliferation stalls, and in some metrics begins to drop. This suppression occurs through a few specific mechanisms. Prolonged estrogen exposure can lead to the saturation and downregulation of local estrogen receptors. Additionally, high estrogen stimulates the release of local cortisol, an adrenal steroid that actively inhibits further cellular division. Prolonged exposure to high estrogen does not result in infinite brain expansion. Instead, The biological system intentionally plateaus its neurogenesis to protect the highly sensitive hippocampal circuits from severe overexcitation. Estrogen does not act in a vacuum. Progesterone acts as a secondary driver, complicating the baseline neural response during gestation. When experimental subjects are treated with progesterone in strict isolation, the hormone successfully enhances the proliferation rate of nerve cells in the dentate gyrus. This chart shows what happens when these hormones are mixed. While estrogen alone produces a high peak in new cell generation, administering estrogen and progesterone simultaneously results in a stunted, strictly moderate level of neurogenesis. The hormonal regulation of brain plasticity is not a simple on and off switch. It is a highly sensitive, competitive balancing act between multiple interacting steroid hormones, constantly limiting and accelerating each other. Producing a high volume of new cells is only the first hurdle. The critical challenge for the maternal brain is ensuring the long-term survival of those neurons. Researchers model the sudden postpartum hormone drop using a protocol called estrogen withdrawal, or EW. EW is an experimental condition that abruptly terminates a sustained estrogen supply to precisely mimic the physiological shock of giving birth. This chart displays the exact consequence of that physiological shock. As the line for estrogen concentration drops steeply, we see a parallel plummet in the survival rate of immature DCX-stage neural progenitor cells. An estrogen-rich gestational environment provides the ideal chemical conditions for initially generating massive numbers of neural stem cells. However, the rapid loss of that specific environment leaves those newly formed cells in a state of profound neurological vulnerability. If the brain cannot adapt its chemistry quickly enough, the postpartum hormone crash acts as a mass extinction event, rapidly clearing out the immature hippocampal neurons before they can fully integrate into the network. The physical and cognitive adaptations required for motherhood rely entirely on a newly born neuron surviving this highly orchestrated burst and subsequent withdrawal of gestational hormones. When these surviving cells successfully mature into functional neuron networks, they actively wire into the existing hippocampal architecture, directly supporting healthy spatial learning and stable maternal behavior. Decoding this precise chemical link between endocrine volatility and hippocampal neurogenesis provides the definitive biological framework for understanding maternal cognitive adaptations, as well as the mechanisms underlying severe postpartum mood disorders.